Behavioral Neuroscience Chapter 8, Segment 1. Sensory receptor organs detect energy or substances. Sensory receptor organs act as filters of the environment. They detect and respond to some events, but not others. The event that affects the sensory organs is called a stimulus. The stimulus can be sound waves reaching the ear, light entering the eye, or food touching the tongue. Receptor cells are within the organ and they detect particular kinds of stimuli and convert them into electrical signals. Eventually, the information from the sensory receptor organs enters the brain as a series of action potentials traveling along millions of axons. Adequate stimulus is the type of stimulus for which a given sensory organ is particularly adapted. Sensory systems of particular animals have restricted ranges of responsiveness. For any single form of physical energy, the sensory systems of a particular animal are quite selective. What type of stimulus was that? Johannes Müller, 1801 to 1858, proposed the doctrine of specific nerve energies, which states that the receptors and neural channels for the different senses are independent and that each uses a different nerve energy. The brain recognizes that different kinds of sensation, called modalities, as separate and distinct because each modality sends its action potentials along separate nerve tracks. Labeled lines is the concept that each nerve input to the brain reports only a particular type of information. Neural activity in one lane signals a sound activity in another. Another signals a smell, and another signals a touch. Sensory processing begins in the receptor cells. A given receptor cell is specialized to detect particular energies to chemicals. Upon exposure to a stimulus, a receptor cell converts that energy or substance into a change in the electrical potential across its membrane. It's called sensory transduction. The devices that convert energy from one form to another are known as the transducers, and the process is called the transduction. The receptor cells are transducers that convert energy around us into neural activity that leads to sensory perception. Some have axons, and those that don't can stimulate associated nerve endings either mechanically or chemically. The initial stage of sensory processing is a change in electrical potential in receptor cells. The structure of a receptor determines the form of energy to which it will respond. The steps between the arrival of energy at the receptor cell and the initiation of action potentials in the nerve fiber involve local changes of membrane potential called receptor potentials. These resemble excitatory postsynaptic potentials. The Pacean corpuscle uses a receptor potential which detects vibrations. It is found throughout the body, in the skin and muscles. It consists of axons surrounded by a structure that resembles a tiny onion because it has a centric layer of tissue. An action potential is generated once the receptor has reached its threshold. It is initiated in its axon terminal. There is a sequence of excitatory events that takes place. First, the mechanical stimulus deforms the corpuscle. Second, the deformation of the corpuscle stretches the tip of the axon. Third, stretching the axon opens mechanically gated ion channels in the membrane, allowing sodium ions to enter. Fourth, when the receptor potential reaches threshold amplitude, the axon produces one or more action potentials. Sensory information processing is selective and analytical. Sensory organs are peripheral sensory pathways, convey only limited information to the brain. Coding. Sensory events are presented in action potentials. Information about the world is represented in the nervous system by electrical potentials in cells. Through some form of coding, a pattern of electrical activity in the sensory system must convey limited information about the original stimulus. It's limited. Each action potential is always the same size and duration. Sensory information is encoded by other features of neural activity, such as the number and frequency of the action potentials, the rhythm in which clusters of action potentials occur, and more. Stimulus intensity. The maximum rate of firing for a single neuron is about 1,200 action potentials per second, and most sensory fibers don't fire more than a few hundred per second. Multiple receptor cells acting in a parallel manner, provide a broader range for coding the intensity of a stimulus. As the strength of a stimulus increases, new neurons are recruited, thus intensify. Range fraction takes place when different receptor cells are specialists in particular segments or fractions of an intensity scale. This requires an array of receptors and neurons that differ in threshold to fire. Some sensory neurons have a very low threshold, which equals high sensitivity, and others have a very high threshold, which equals lower sensitivity. The clue to the intensity is whether it activates only low threshold receptors or both high and low threshold receptors. Stimulus location. Some sensory systems reveal location by the position of excited receptors on the sensory surface, which is most evident in the somosensory system. In both the visual and touch system, cells at all levels of the nervous system form surface sheet of receptors to the cerebral cortex are arranged in an orderly map-like manner. The map at each level is not exact, but it reflects both the positions and the receptor density. More cells are allocated to the spatial representation of sensitive, densely innervated sites like the lips or the center of the eye. With bilateral receptor systems, which is your two eyes, your two ears, the relative time of arrival of the stimulus at the two receptors, or the relative intensity, is directly related to the location of the stimulus. As the stimulus moves left or right, the receptors of the left and right sides are more excited asymmetrically. Adaptation. Receptor response can decline even if the stimulus is maintained. Adaptation is the progressive loss of receptor sensitivity as stimulation is maintained. It is demonstrated by recording action potentials in a fiber leading from a sensory receptor that is receiving a constant level of stimulations. There are two kinds of receptors. Tonic receptors show little or no decrease in the frequency of action potentials as the stimulus is maintained. They show little adaptation. Phasic receptors display adaptation, rapidly decreasing the frequency of action potentials when the stimulus is maintained. Adaptation means that there is a progressive shift in the neural activity away from the accurate portrayal of maintained physical events. Sensory systems may fail to register neural activity even though the stimulus continues. Sensory systems emphasize change in stimuli because the changes are more likely to be significant for survival. Sensory adaptation prevents the neuron systems from becoming overwhelmed by stimuli that offer very little news about the world.